Can somebody confirm you can hear me?
We can hear you.
Thank you. All right, before we begin, are there any questions? If not, let's begin. Today is January 16th, and we're supposed to be covering Chapter 2, Chemical Principles. Any question about that? Does anybody have a problem with Respondus Lockdown Browser? I haven't checked my emails, so. All right, let's go on with Chapter 2. There's no questions, no comments. I have that we were talking about the polysaccharides in our last slide. Any questions about the polysaccharides? If not, let's move on to lipids. People are still logging in here. Let's see how many people I have here. I can't see it. Come on. 23. Well, that's pretty good. All right. Lipids consist of carbon, hydrogen, and oxygen. However, there's very little oxygen in lipids. They do have a little bit, but there's very little oxygen in lipids. I'll show you a slide in just a minute. Lipids are mostly nonpolar and insoluble in water, meaning lipids tend to be hydrophobic. There is one exception, we'll talk about that. But other than that, lipids are nonpolar and insoluble in water. They are structural molecules and play energy storage roles in living cells. Lipids are primary components of cell membranes. We have lipids of three different types: simple lipids, complex lipids, and steroids. You could have an author that will break them into more types. Like some people will break the waxes into a different thing, and what else? There's five lipids that other authors use. I don't remember what the fifth one was, but we're going to use the textbook. It breaks it down into simple lipids, complex lipids, and steroids. Fatty acids are a major component of lipids, at least most lipids, not all lipids. They consist of chains of carbons, meaning a hydrocarbon chain. They do have a carboxyl group on it, COOH. It's at one end of the molecule. Let me blow this up. So there's a fatty acid molecule with COOH, and you can see this molecule is mostly carbon and hydrogen. What most lipids are carbon and hydrogen. They do have a little bit of oxygen on them, like right here. You have two types of fatty acids: a saturated fatty acid, shown here, and an unsaturated fatty acid. A saturated fatty acid has all of the carbons linked together with a single um, covalent bond linking the carbons together. And of course, they have single carbons linking the hydrogen together because the hydrogen can only bond to one atom. But the carbon can bind to four different atoms. And if it has only single carbon chemically covalent bonds linking the carbons together, it's a saturated fatty acid. So this fatty acid is a saturated fatty acid. An unsaturated fatty acid has at least one double carbon bond linking two carbons together. And that we call an unsaturated fatty acid. Wherever there's an unsaturated link, it'll cause a kink in the hydrocarbon chain, which I've got, I believe, in the next slide here. And blow this up a little. So right there, we have an unsaturated fatty acid with a double carbon bond, both chemically two carbon covalent chemical bonds, linking the carbons together. And where that happens, we get a kink in the molecule shown here. These are saturated fatty acids, and this is a fat or a triglyceride because there's three fatty acids hooked to a glycerol molecule. And if there's only one unsaturated fatty acid in the molecule, you call this entire molecule unsaturated. Any questions about any of that? So what you do is you have a fatty acid shown here and a glycerol shown here. The water can come off, the hydrogen on the glycerol, the OH on the carboxyl group of the fatty acid comes off, forming water that will link this carbon to that oxygen shown there. And that's how a fatty acid can combine with a glycerol to make a triglyceride. A triglyceride has three fatty acids linked to the glycerol, one in each position. And you only need one unsaturated chemical covalent bond between the carbons to make this whole molecule called unsaturated. If it was saturated, then all of the carbon links have to be a single carbon chemically covalent bond linking the carbons together. Now, as you know, saturated fatty acids or saturated fats or saturated fatty acids are not as good for your heart as unsaturated fatty acids. And that's the only difference in a fat. There'd be one chemical double carbon bond in the covalent bond, making this an unsaturated fatty acid. Any question about any of that? And
I
surprising,
have a question.
go on.
I have a question. So, why does the carbon to carbon double bond cause a kink, but then the oxygen to carbon bond doesn't cause a kink?
Are you talking about this double bond there?
Uh,
That's where
the
this is not a
bonds in
double
the,
bond. That's
I'm
not a
talking
double
about the
bond.
double
That's the
on the
only
right
double
side.
bond with the oxygen right there. That's not linking with anything, so it can't cause a kink. This
I was
bond,
talking about on the right side, the carbon to carbon one, where the kink is.
uh-huh.
I was wondering why that causes a kink.
I'm not sure why that causes a kink in the molecule, but here would be an unsaturated um, carbon, and it's just a straight carbon chain having a hydrogen. Whenever there's only two hydrogens and there's a double carbon covalent bond, it causes a kink. And I'm not exactly sure, I'm not a chemist, why that causes a kink, but it has to do with the fact that um, it's a double carbon bond and there's only two hydrogens, one each hydrogen to each carbon, and it causes that to kink. Um, I can't explain why because I'm not a chemist. I just don't know.
Okay, thank you.
All right. But it does cause a kink there, and that's all you need to know. Um, I'll have to look up why that's a kink and maybe get back to you, but I don't know. All right, let me shrink this down. Okay, fats shown here, or a triglyceride. A triglyceride is the same thing as what we call a fat, are the body's richest energy source. And the body stores fats as adipose in animals. And so if you needed a high energy source, let's say your car breaks down in Nome, Alaska, that's way northern Alaska, and it's in the wintertime and you got 20 miles to hike into town and you have food in your car and you only can carry, let's say, 20 pounds because you got to walk into Nome. And the thing to carry to give you the most energy per weight of the food that you're carrying would be a fat. because it's the body's richest energy source. We get more energy from digesting a fat molecule than you would from carbohydrates or protein or any other molecule.
Can I add something real quick? The
Uh-huh.
reason why it makes a kink is because the hydrogens are both on the same sides, and they call that a cis configuration.
This is a cis, but I think if you have them trans, it still causes a kink.
Gotcha. I'm just saying in that one, generally, if the
Yeah.
hydrogens are both on the same side, it'll cause a kink. That's all.
Okay. Yeah, I'm not sure why there's a kink, but I think there's a kink in the trans too. All right. Let me see if there's anything else. Triglycerides are the most common dietary lipid in our diet. It's the most common fat in our body, most common lipid in the body. And to see some triglycerides, just look at your belly. You got lots of triglycerides there. Even if you're thin, everybody has triglycerides in their belly. In fact, humans have triglycerides underneath their skin. We, I don't know, store it there. All right. That's enough for fats and fatty acids. I guess forgot to mention, simple lipids are simply a combination of fatty acids and glycerol to make the simple lipid. The triglyceride is the most common one where you have three fatty acids linked to the glycerol. So simple lipids are a polymer made up of three molecules, or at least triglycerides are, including glycerol and then three fatty acid components. These are the monomers, and then triglyceride is a polymer. There are only some lipids which are made up of monomers. The triglyceride is the first, and generally simple lipids are that way. The second group of a simple lipid is a phospholipid. It too is a polymer made up of monomers. Let me blow this up a little bit. The glycerol is right here, and it has two fatty acids linked to it. There's one there, and there's one there. And then it has on the third portion of the glycerol a phosphate group to it. Phospholipids are unusual because this is actually a charged molecule, and the phosphate part as well as the glycerol part are, what do you call that? They're polar. So this side of the phospholipid containing the phosphate is polar, and this side of the phospholipid is nonpolar. So the fatty acids are nonpolar, and I didn't mention it, but I did mention that most lipids are nonpolar. The simple lipids and the fatty acids are nonpolar. So a triglyceride is nonpolar. The phospholipid has a polar region, the phosphate portion, which we call the head. This portion here is called the head. And then the fatty acid portion are called the tails of the phospholipid. The tails are nonpolar. This makes this molecule biphasic, which is an unusual molecule. That means it's biphasic. It can dissolve in water because of the polar group, and it can dissolve in lipids like oil because of the nonpolar group. Any questions about any of that? Bipolar molecules are not very common, but you do know one, or two maybe. Soaps and detergents are both bipolar, and that is why soaps and detergents help us clean our clothes. Because if you have a water-soluble molecule in your clothes and you want to wash it out, well, you don't even need the detergent because the water will pull out the polar molecule from your clothes. But if you have a lipid in your clothes, like the lipids from your skin, the water will not pull it out. And then we put detergent, which is a bipolar molecule, having a portion that is polar and a portion that's nonpolar, which phospholipid is an example of a bipolar molecule, but it is not a detergent or a soap. But the point is that the nonpolar region of the molecule can dissolve in the lipids in your clothes, and then the polar part of the molecule can dissolve in the water. And that's why detergents help us clean our clothes and why we add detergent to our laundry when we're putting it in the washing machine. Any questions about any of that? All right. So a phospholipid is unusual because one portion of the molecule can dissolve in water. The other portion can dissolve in lipids or oil. Phospholipids are the main molecule in a cell membrane. And when you get lots of phospholipids together, they will start arranging like this in a body of water. And they could be like that or like that. And then they will then circle around on each other, forming this or that. You don't need to know about this, but can form a liposome, which will have water on the outside of the liposome and then water on the inside. What does this look like to you guys? Should be familiar. What looks like this?
An eyeball?
That is kind of close, but you have to realize that this is cut in half. So you can make that a full circle so that you can see this portion will be on the outside and then this will be on the inside. And that's the bilayer composing the phospholipids. And then the outside there will be water. What does that remind you of? So this is not really an eyeball. It's something else. It is biological.
Skin.
Kind of. I've got it right here. It's a cell. It should remind you of a cell. This would be the inside of the cell, which would be the cytoplasm. And what's the cytoplasm mostly made of? Water. And then this would be the cell membrane, which will have a phospholipid with the head facing inside the water, and then the non-polar parts of the tails of the phospholipid on the inside of the membrane, and then the heads of the second layer of the phospholipid facing outside the cell, because outside the cell in a body is mostly water. Think of this being like a red blood cell in the blood. Obviously, the blood is mostly water. It doesn't matter where you are in the cell. Outside the cell is mostly water because the body is mostly water. Any question about any of that? All right. So as I stated, phospholipids are the main molecule in the cell membrane. Now, in animals, which do not have a cell wall, animals have cholesterol. Let me blow this up. Cholesterol in their cell membrane. So that's cholesterol there and there and there. And the cholesterol gives strength and support to the cell membrane. The reason being that animals don't have a cell wall to protect the cell membrane. And so animals put cholesterol in their cell membrane to give it strength and a little bit of more support, stronger than the phospholipids alone. And there are other molecules in the membranes. We don't have them shown, but obviously there's proteins and carbohydrates. But the main molecule in the cell membrane is phospholipid. And then I guess the second main molecule in animals would be cholesterol. And I'm not sure what the third would be, probably proteins, because we have a lot of transport proteins in the cell membrane, as well as other proteins. All right, any question about any of that? All right. So let's move on to our third group of lipids, and that is a steroid. A steroid is a four interconnected rigid carbon ring molecule. So there's a steroid with 1, 2, 3, 4 rings, and there can be other parts of the molecule. If there's an OH group attached to a steroid, we call it a sterol. And this has an OH group attached to it, so it is a sterol. This molecule is specifically cholesterol, which, as you can see, is inside the cell membrane of this cell membrane. And you only find cholesterol in animals. So if you want to have a cholesterol-free diet, you don't eat anything that you get from animals, meaning any animal product. I suppose if you had skim milk, that would be having no cholesterol in it, but any other milk would have cholesterol in it. And skim milk, if you know, has the cholesterol in it. They just remove all the fat, including the cholesterol, and they make it skim milk, and that's why it doesn't have any cholesterol in it. Any question about any of that? So plants do not have any cholesterol. So if you have a vegan or vegetarian diet, that's all vegetables or all fruit, meaning all plant-based. And I suppose if you were to eat produce too, I can't think of anything what you could eat like that. If you were to eat algae, that would be a produce. That would not give you any cholesterol. To get cholesterol, you have to eat animal products, mainly red meat. Okay, so here we're seeing cholesterol. Is this molecule made up of monomers? Or is it just a one molecule? Trying to make a point here. All right, the phospholipids and the simple lipids, like a triglyceride, are made up of monomers. There's one there, there's one there. Here's one here, there's two there. These are monomers making up the polymer. Here's the molecule there. Where's the monomer? Anyone? There is no monomer in a steroid or a sterol, meaning this is a lipid that is not made up of monomers. Any question about that? So of the four biological molecules, only lipids have some lipids that are not made up of monomers. All of the other four biological molecules, meaning all the proteins, all the carbohydrates, all the nucleic acids, they are made up of monomers. And of the lipids, some of them are made up of monomers and some of them are not. All right, any question about lipids? No questions? Let's move on to proteins. Proteins are essential in cell structure and functions. Proteins may be structural proteins found in bone, muscle, hair, meaning structural parts of the body. Proteins can be enzymes. Enzymes speed up chemical reactions. Enzymes can be transport proteins that move molecules or chemicals across a membrane, like into the cell or out of the cell. Proteins can be regulatory proteins that regulate body functions. Example, hormones, like a growth hormone in a child will cause the child to grow. Proteins can be responsible for movement, like the movement of my hand, assuming you can see this, is happening because of a protein. And then when we walk, or when a fish swims, or a cell swims, it's because of the protein, the movement of the protein. Proteins can also be responsible for some bacterial toxins. So if you eat something and then get food poisoning because of a toxin in the food caused by some microbe, that could be because of a bacterial toxin. There are some toxins which are not proteins, but the majority of them are proteins. And then there are some proteins that we call antibodies. These are proteins that animals make to help defend the body from foreign infection. Any question about any of that? Well, did you note that proteins serve a lot of functions, and they're different functions in an organism or even in a cell? That could be with the cell, 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 that could be with the cell. That could be with the cell. That would not be with the cell unless we're talking about a B cell like of a person. But all the others can be found in every cell, if the cell moves, of course. Any question about that? Well, do you notice that proteins are more diverse and serve more functions than any of the other biological molecules? That's simply a yes or no question. Hopefully somebody will answer.
Yes.
Yes, they are. They are more diverse. The reason why they are more diverse is because they have more monomers making up the macromolecule, the protein or the polymer. And that makes proteins more diverse than any of the other biological molecules. Meaning there's a handful, maybe possibly up to 10, 15 different monomers of carbohydrates. And most carbohydrates in the body are made up of simply probably about 3 or 4 different monomers, but you can go higher. And then the lipids in the body, if they're made up of monomers, there's only a couple of monomers that make up the body, although this one certainly goes to 12 or higher. And then in nucleic acid, each nucleic acid is only made up of 4 monomers. But with the protein, each protein is made up of 20 different monomers. And that we call 20 different amino acids. So that makes proteins more different than all the other biological molecules because they have more monomers. So all proteins consist of monomers. We call these monomers or subunits an amino acid. Here's the generalized shape and function of a amino acid. It has a central carbon that all other portions of the molecule bind to or indirectly bind to. It has a hydrogen on one side of that carbon. It has a amino group attached to the central carbon on one side of the molecule. And this is a functional group giving the molecule different properties. And on the other side, opposite of the amino group, it has a carboxyl group also attached to that central carbon. This is a functional group giving the amino acid properties. Two of these properties, both of them have, this is a polar group, and that's a polar group. And so all amino acids have polar groups to it, meaning all amino acids can dissolve in water. Any question about that? And then the fourth group attached to that central carbon is called an R group. The R group can vary depending on which R group we have. It makes up the amino acid. Like for tyrosine, this would be the R group of tyrosine. And humans have 20 different R groups because we have 20 different amino acids, each different amino acid having a different R group. Any question about any of that? All right. You do not need to memorize the R groups. I'm just going to show you some of the 20 different R groups. The amino acids only differ in that R group. And all of these amino acids, meaning this hydrogen, that, what is that called? Methyl group there. These is a chain of methyls groups. That's another bigger chain of methyl groups. That's a chain of methyl groups. Branch chain, branch chain. Well, it's sort of a branch chain. An unbranched chain, but that's only one. Another unbranched chain would be if you simply had the methyl groups hooked one to another in a straight line. All of these amino acids are nonpolar in their R group, meaning all of these R groups are nonpolar. And that means that some amino acids are also nonpolar from their R group. Like all of these are nonpolar here, but remember they're polar in their amino group and their carboxyl group. So they always have a polar group, and some amino acids have a nonpolar group if their R group is nonpolar. Some amino acids have a R group which is polar or partially polar, like this OH group is polar, making this R group at least partially polar. This part of the R group is nonpolar, but that part is polar, so that this part can dissolve in water. This is polar on the NH group here and the NH group there, making this R group also polar. That R group is nonpolar, and that R group is nonpolar. Any question about any of that? The simplest R group, I didn't mention that, is simply an H, and that is the amino acid glycine. You don't need to know that. I'm just pointing it out, that the R group makes the different amino acid, and they can be different groups in the amino acid, one different for each amino acid. Any question about any of that? Like I said, do not memorize these different amino acids. I'm not going to quiz you on that. But humans do have 20 different amino acids. They only differ in the R group. Most living things have 20 or very close to 20 different amino acids. So most living organisms have about 20 different amino acids. And if they change an amino acid, meaning let's say they don't have alanine, we'll put in something else, another R group there. And then once again, that will be the life form will have 20 different amino acids. The highest that biologists have found, you don't need to know this, there is a bacteria that the species has 23 different amino acids. And that's the highest that I know of, that I've heard of. I don't know what the lowest is, but it's close to 20. And most living things have 20, the same as humans. But they may not have the same amino acid as humans. Any question about that? All right. Amino acids are linked together by a peptide bond. So one amino acid has the OH come off of this amino acid and the H come off of that amino acid. And then that carbon over here binds directly to that nitrogen right there by dehydration synthesis, meaning water is formed. This bond is unusual in living organisms, as well as it's unusual in the universe. Nitrogen does not often bond chemically to carbon. And so we call this bond a peptide bond because we find it in peptides or proteins. If you don't know, a peptide is a molecule of amino acids that's shorter than a protein. And then you just link the different amino acids together to make the protein. And that is how a polypeptide chain actually forms. And when you add the last amino acid to the polypeptide chain, that's when we call it a protein. So the peptide bond is rare in nature, even in the universe, but we do see it in amino acids forming peptides or polypeptides or proteins. So a polypeptide is just a chain of amino acids linked together by a peptide bond. A polypeptide that has all of its amino acids added, meaning the last amino acid is added, we then call that a protein. So as a protein is growing, you need to call it a growing protein, or you call it a polypeptide. And not until the last amino acid is added do you call it a protein. But you could call it a growing protein because you're adding the amino acids. Any question about any of that? All right. So how the body makes proteins is they just link amino acids together by dehydration synthesis, linking one amino acid after the other, creating a straight chain that we will eventually call a protein. Let me blow this up. There are 4 levels to the structure of a protein. So 4 levels of protein structure. When the last amino acid is added to the growing protein, we then have the primary structure of the protein. All it is the sequence of amino acids, one after the other. After that, we have the secondary structure of the protein. That happens when the nearby amino acids start interacting, and then the protein starts beginning to fold. The tertiary structure of the protein is when amino acids from all over the protein can interact with other amino acids all over the protein. That will be where the protein takes on its knot shape. What used to be a single line in the primary structure. In the tertiary shape, it takes on its complex 3-dimensional shape, like that. And the last near an amino acid near the end of the protein can combine with a amino acid near the start of the protein, meaning it's amino acids all over the protein can interact with other amino acids anywhere on the protein. And that's what we call the tertiary level of the protein structure. Some proteins never go higher than the tertiary level because they only have one polypeptide unit forming the protein. So all enzymes that I know of only go as high as the tertiary structure because all enzymes only have one polypeptide forming the finished protein. But some proteins can have a quaternary level, and these will be when different subunits or different polypeptides come together to form the final protein. Like an antibody is made up of 4 different polypeptides that come together to form the antibody, the protein. And when all 4 of them come together and take on their shape, that we call the quaternary level of the protein structure. Any question about any of that? All right, let's watch a little video discussing the different levels of protein structure.
How much money do you spend
Commercial
per month
here.
living in New York City, including everything?
8K the last 2 months.
And what's your occupation? What do you do?
I do.
There we go.
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well.
I've already talked
The
about
amino
that.
acid is the N-terminal. Now, these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. Hydrogen left over. It's a di. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure.
All right, let me interrupt here and say that he's only talking about the hydrogen bonds in the secondary structure. Let me say the secondary structure can be stabilized by other bonds like ionic bonds, or it could be, I don't know, stabilized or whatever, formed by repulsion as well as attraction. And one repulsion would be if you have a nonpolar amino acid here and a polar amino acid here. These 2 amino acids will be repulsed from each other. And then if this is a hydrogen bond, obviously that's a polar molecule and that's a polar molecule, that will be an attraction between 2 amino acids. Another attraction would be if you had an amino acid with a negative charge and an amino acid with a positive charge. That would be causing an ionic bond between them. And that would be also a way that the secondary structure could be stabilized. So the hydrogen bond is probably the main bond stabilizing the secondary structure, but there are other, not bonds, but there are other forces that can stabilize the secondary structure, and they could be forces of attraction, like the ionic bond, the hydrogen bond, or even another one that is an attraction is the van der Waals forces, and that is if this is a nonpolar amino acid, and this is a nonpolar amino acid, they will like being together. and how you can visualize that. When you put oil molecules together in a bottle of oil, the reason why the oil forms a liquid is because the oil molecules are held together by van der Waals forces. These are nonpolar molecules, like to be around other nonpolar molecules. It's a much weaker bond than the hydrogen bond, but it is still well, not really a bond, it's an attraction between two nonpolar molecules. Any question about that? I'll talk a little bit more about that, but my point is, he's only talked about one force that puts this secondary structure in there, and there's several forces that come into play to make the secondary structure. Any question about any of that? What's unique about these forces making the secondary structure is, let's come back to here. The secondary structure only comes about from amino acids that are nearby each other, that interact with each other. It does not happen from an amino acid near the end of the protein and near the beginning of the protein, because these amino acids have to be nearby each other to form the secondary structure. Any question about any of that? All right, let's talk about the tertiary structure. There we go.
Next up, we have the tertiary structure, which represents the three-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
Are you struggling in your calculus?
All right, any question about the movie video 4 levels of protein structure? If not, let's go on. The primary structure, once again, is the sequence of amino acids in a polypeptide chain. So once a protein is made, all it has is the primary structure where there will be one amino acid following the next. Any question about any of that? You only see the primary structure really when the protein is first made. It has the primary structure whenever you see a protein, but my point is when it's only the primary structure, that's when the protein is first made. Once the protein is made, the nearby amino acids will start interacting with each other. And here we can see right there, that's forming a hydrogen bond right there. And this will be a polar molecule being attracted to another polar molecule and then a hydrogen bond forming. That is one force of attraction. But we could also have a positive charge on this amino acid and a negative charge on that amino acid. And that will be a force of attraction between these two amino acids. You can also have, like between these two, there's no bond there, so there's no hydrogen bond. These could be two nonpolar amino acids. Nonpolar amino acids can be attracted to each other because of van der Waals forces, meaning a nonpolar molecule likes to be around another nonpolar molecule. It's a very weak attraction. We call it van der Waals forces. Hopefully you had that in the either in chemistry or in your biology 162 class. If not, I'm mentioning it here that nonpolar molecules can be attracted to each other by a very weak attraction called van der Waals forces. And the example of that is why our molecules of oil form a liquid in our bottle of oil because of van der Waals forces. And the van der Waals forces break and reform all the time why the bottle of oil is a liquid. But if you were to heat it up, the oil would become a vapor. And there, the van der Waals forces cannot hold the molecule together, hold the liquid together. And that's why it's a vaporized liquid, same as water. But the difference is water is held together by hydrogen bonds. Oil is held together to form the liquid by van der Waals forces. Those are the forces of attraction in the secondary structure. Just checking to make sure I got all of them. There are also forces of repulsion, like right here and right there. These two amino acids are being repulsed from each other. It could be this one has a positive charge and that one has a positive charge. That will be repulsed from each other because two positives don't like being around each other. They'll be repulsed from each other. Another force of repulsion could be, this could be a nonpolar amino acid, and that could be a polar amino acid. They will be repulsed from each other. Let me see if there's any other force of repulsion that I can think of. That's all the forces of attraction and repulsion that I can think of. And all these forces of attraction or repulsion can make the secondary structure where nearby amino acids are interacting with each other. Forces of attraction here, 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 and there. Forces of repulsion right there. So this is the same amino acid. So that would mean that this force of repulsion means both of these probably have a positive or both have a negative charge because it's the same amino acid. Any question about that? Now, the video told you that you need to know about the alpha helix and the beta pleated sheet. Those are just two secondary structures. It did show you the third secondary structure. Just never said what it was. There we go. And that's the random coil, where the molecule just coils randomly. Like this right here would be a random coil. And there would be forces of attraction right here between this amino acid and that amino acid. And that's why they're together. I'm going to have a hard time drawing the hydrogen bond trying. Sort of. Well, that's pretty good. You can have forces of attraction, like this could be a positive amino acid, that could be a positive amino acid. The random coil, I don't only mentioning it because it's kind of helpful to mention with the tertiary structure. In the tertiary structure, I guess I should put something right here, where there's forces of attraction, forces of repulsion between this group here and that group there, and here, this amino acid here, and that amino acid here. And then I guess that amino acid there, and that amino acid here. And that's why they're so close together right there. This would be a force of attraction, but you notice that this amino acid is near the beginning of the polypeptide or the protein. And this one, let's shorten this. Get the eraser here. This is near the end of the protein. So this is not a secondary attraction. This is the third tertiary attraction, third level of protein attraction. And then the third level of protein attraction or whatever, the amino acids from all over the protein can interact with other amino acids in the protein. Let me go back and do something right here. Put this one out this way. Okay. And there you can see this one is being repulsed from this amino acid here. And that's why it's not quite right about there. So repulsed from that one. And that's why this is not close together like this one and this one. These two are forces of attraction there. Okay, so there can be forces of attraction and forces of repulsion forming the tertiary level as well. Any question about that? So this is the third level. Now, the forces of attraction and repulsion can be the same forces we saw in the secondary level. Oops, didn't spell that right. There's one different force of attraction that we see in the tertiary level and you do not see in the secondary level. These are the disulfide bridges. Which form between amino acid cysteine. So let me make this cysteine right there. I can get that up a little. So that amino acid, well, let me copy that actually. Almost down. Not able to get it. Let's see if this gets it. Yeah, I got it. So two cysteines. That cysteine here. And this cysteine here. They have a hydrogen group on them, which I'll have a hard time drawing that in here, but we'll just put a sulfur. Trying. So this will have a sulfhydryl group, and that will have a sulfhydryl group. I'm going to have a hard time doing this because I don't have enough space in there, but you're getting it. And what happens is the hydrogen comes off, and the hydrogen comes off, and now we have a hydrogen bond forming. Oop. Third one in. Delete that. Come on. There we go. So there is a sulfur covalent bond now between this cysteine and this cysteine, and the hydrogen just come out into solution. So let me go off over here. Oops, that's the wrong one. The hydrogens just come off and go into solution. Just going to put that like that. Trying to draw a plus here. I don't know if I can do it. Not too bad. Kind of a plus. And then the cysteines linked together by a disulfide bridge, where this sulfur of this cysteine and this sulfur of this cysteine actually link together. That makes a very strong interaction between this cysteine and this cysteine, because that is a chemically covalent bond. Much stronger than a hydrogen bond shown here. Any question about that? And if you don't know, when you get your hair permed, what they do is they form disulfide bridges between cysteines in your hair. And that's why when you walk into the shower, you can wash your hair about for a month, and that cysteine disulfide bridge will last for about a month when you get your hair perm. So this is a very strong bond. Whereas if you don't know, you get your hydrogen bond in your hair. My mother used to curl her hair. She would get a curling, I don't remember what the device was called, curling heater, and put her hair in a curl and hold her hair around the curl to make the curl and put it in a bobby pin. And then she'd do this all over her hair. And then she would put the cap on, turn on the machine, which I think put heat, maybe a little bit of moisture in the hair. And then she would be there. I don't know for how long, I don't remember. It was maybe 10 minutes, maybe 15 minutes. And then she would turn the machine off, take off the I don't remember what that was called, the hair bonnet, and take off the whatever that pin was called you put in your hair, took off the curling iron, and then she would let her hair out and it would have this curl. And then she would go out. She lived in Western Oregon. She'd go out in the Western Oregon mist or the rain, and then that curl would sag immediately because the hydrogen bond formed by the curling iron was very weak, and any moisture would break that hydrogen bond, forming the curl. But if she would have gotten her hair permed, which back then that they didn't have that, but later on they did. I'm talking about way back in the 1960s. My mother didn't curl her hair in the 1970s or the 1980s or any time after that. But and I was only a child in the 1960s. But with the perm, you form the sulfur disulfide bridge between the cysteines, and that's a very strong chemical bond. And that perm of your hair will last for about a month, taking showers at regular intervals. Any question about any of that? My point being, the third level of protein structure has all of the forces of attraction and repulsion as the second level of protein structure, but it has one additional force of attraction, and that's the disulfide bridge, which is a very strong level of attraction. Any questions about that? All right. The tertiary structure, let me remind you, forms the three-dimensional structure of the polypeptide unit. And if there's only one polypeptide unit to the protein, it forms the three-dimensional structure of the protein. And the amino acids can interact from all over the protein, like this amino acid interacting with that amino acid. This one's near the end of the protein. This one's close to the beginning of the protein. And remember, in the secondary structure, you can only have amino acids nearby each other, interacting with each other. All of the forces of attraction and repulsion in the 3D structure are the same as seen in the secondary structure, with one difference, and that is the disulfide bonds that can form between two cysteine amino acids. Any question about any of that? There's a mistake. All right. If the protein only is made up of one polypeptide, the tertiary structure is the highest level of protein structure that that protein can get. To my knowledge, all enzymes only can go to a tertiary structure because all enzymes that I know only have one polypeptide unit making the enzyme. But there are some proteins that have more than one polypeptide unit making the final protein. And antibodies are made up of four polypeptides. Hemoglobin is also made up of four polypeptides to make the final protein. In the quaternary structure, all of the polypeptides have to come together, and there will be amino acids interacting, like these amino acids here are being attracted to the amino acids of this polypeptide, and then this amino acid here will be repulsed from that amino acid, and that's why they're not together. You find the same forces of attraction in the quaternary structure that you do in the tertiary structure, meaning the disulfide bonds can be one of the forces of attraction between the different polypeptides. Quaternary structure is only seen on some proteins that are made up of more than one polypeptide subunit, such as hemoglobin and antibodies. They're made up of 4 polypeptides. Insulin is also another protein that's made up of more than one polypeptide, so it has a quaternary structure. Any question about any of that? So not all proteins have 4 levels of protein structure. Some proteins only go as high as 3 levels, and that's probably most proteins. All right, any other questions about the different levels of protein structure? A little more, we're going to talk about proteins. You can discuss the overall shape of a protein, and we say proteins are globular if the protein is roughly spherical. Protein is compact. Most enzymes, actually all hemoglobin, we call them a globular protein. You can have a protein which is fibrous, and that protein is thread-like. Collagen is an example of a fibrous protein. Some structural proteins are fibrous. We also have something that we call conjugated proteins, and this is where the protein is combined with an organic or inorganic compound. Could be an amino acid, could be a protein. It's bound to something else. We call it a conjugated protein. The protein is named by their non-protein component. Like a glycoprotein is a protein bound to glycogen, meaning a carbohydrate base unit, and that's a glycoprotein, which is found in the blood. We also have lipoproteins, which are a protein bound to a lipid, and there are lipoproteins also found in the blood. Any question about any of that? When we're talking about proteins, if the protein has its 3-dimensional shape that it's supposed to be in, then the protein can be functional. But if the protein starts to lose its 3-dimensional shape, breaking hydrogen bonds that should be there to hold the protein together, forming hydrogen bonds that shouldn't be there, like when you put protein in the refrigerator, the protein can form hydrogen bonds that shouldn't be there, changing the 3-dimensional shape of the protein. That loss of the protein function, we say the protein has begun to denature. And when the protein has totally lost its 3-dimensional shape, can no longer function, we say the protein is totally denatured. So there's partially denatured and then fully or totally denatured. And usually they just use the name denatured to mean fully denatured. A protein can start to denature if it's at its incorrect temperature, its incorrect pH, or its incorrect salt concentration. Any question about any of that? All right, we'll talk a little bit more about protein denaturation, I think, in a future lesson. But for now, all you need to know is that proteins have to be in a certain environment. They have to be at the correct temperature, the correct pH, and the correct salt concentration if the protein will function at its optimal level, meaning its best rate. And if you change the temperature, the pH, and the salt, the protein will not function at its optimal level. And if you change it far, the protein will not function at all. All right, any question about any of that? Let me briefly talk about nucleic acids. Let me check this here. Nucleic acids are the fourth biological molecule. There are two types of nucleic acids you must know, and that is DNA and RNA. Those are the two nucleic acids. DNA, its long name is deoxyribonucleic acid. RNA, its long name is ribonucleic acid. Each DNA and RNA consist of monomer units called nucleotides. A nucleotide consists of three parts: a pentose sugar, shown here. It's a 5-carbon sugar, which can be either deoxyribose in DNA or ribose in RNA. It has a phosphate group, shown here, and then a nitrogen-containing base, shown here. Those are the three parts of a nucleotide. And each nucleic acid, either DNA and RNA, is composed of monomers that we call nucleotides. DNA has 4 monomers, and RNA has 4 monomers. So DNA is only made up of 4 monomers. RNA is only made up of 4 monomers. Let's move on. In DNA, we make the DNA out of the monomers adenine, guanine, cytosine, and thymine. And these are often abbreviated A, G, C, or T. You need to know either name. You don't need to know both names, but you need to know either adenine or A, guanine or G, cytosine or C, thymine or T. And I use them interchangeably, so live with it, I guess. The bases are adenine, guanine, cytosine, and thymine. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. How you can remember these is a pyrimidine has a Y in it. Cytosine and thymine each have Y's in it. Adenine and guanine do not have a Y in it. So adenine and guanine are not a pyrimidine, they are a purine. Any question about any of that? And then for RNA, it is composed of 4 nucleotides as well. A, G, and C are the same 3, but it does not have T, thymine. Thymine is replaced by uracil. You just need to know that uracil replaces thymine in RNA. Thymine has a Y in it, so thymine is a pyrimidine. Uracil replaces thymine, so uracil is also a pyrimidine. Any question about that? The bases are important because DNA has a pairing where one strand of DNA will pair to another strand of DNA. We call that complementary base pairing. A in DNA will only pair with T, and T only with A. And C will only pair with G, and obviously C with G with C. Okay, this is complementary pairing. A never pairs with C, never pairs with G. And that's for DNA. For RNA, remember we don't have T, we have U, and U pairs with A. You don't really need to know that because RNA most of the time does not pair with something. Okay? The only time it ever pairs is when RNA is first being made. It actually begins to be paired with DNA. And we'll talk about that when we're talking about RNA synthesis. So you don't need to know that now. Any question about that? How that pairing occurs is A, hydrogen bonds with T, and A and T form two hydrogen bonds between the A and the T. We go to the next slide. C and G also pair with each other. Blow this up. But they form three hydrogen bonds between the G and the C. Any question about any of that? If not, I think I will stop here, and I will see some of you in the lab at 6:30. I'll see the rest of you in the lecture next week. Any questions before I go? All right, goodbye.